everyone. My name is Kacie Deters. I'm an assistant professor at UCLA in Integrative Biology and Physiology Department. I've been with the Bruin family since July of 2022, and I'm very happy and thankful to the organizers for allowing me to share some of my research that I have done and that I will be doing while at UCLA. So first I want to talk about Alzheimer's disease, which is the most common form of dementia. The two hallmark pathological features of AD are extracellular amyloid plaques and intracellular tau tangles. Another key characteristic of Alzheimer's disease dementia is progressive cognitive decline, as shown here on the x-axis. Some cognitive decline is completely normal as we age, which is shown in the green line above. However, before overt dementia is present, individuals might show brain pathology of those proteins like amyloid and tau before cognitive symptoms are apparent to the individual or those around them. This is termed the preclinical stage. As time progresses, cognitive impairment starts to become apparent to the individual or those around them, and the person declines to dementia as neurodegeneration continues over time. So we have this dotted line here that kind of separates this preclinical stage before symptoms are present, and then the mild cognitive impairment or MCI stage and dementia stage when symptoms are present. So we can use something called biological markers or biomarkers in order to track these different stages of Alzheimer's disease dementia. And it is very important to note that the different stages of Alzheimer's disease do not exist as discrete units. They do exist on a continuum, as shown here in this figure. If you look at the x-axis, we have those cognitive stages I just talked about in the previous slide, normal preclinical MCI dementia on this continuum. So in the preclinical stage, again, we have these protein abnormalities that can start to be measured. So for example, you have amyloid that is usually apparent in the brain up to 20 years before any dementia symptoms are present. And then even tau, as shown here in the green line, is present. And then as time progresses, there are other biological markers that start to become apparent. So for example, we can use MRI or look at different anatomical features, and we start to see that brain structure, changes in the brain structure. So for example, the hippocampus is really important in memory. It's located in the atrophy in people as they get closer to the MCI stage. Again, cognition typically is not apparent until the MCI stage when it's mild cognitive impairment. So the name really matches when you start to see different abnormalities on the neuropsychological assessment testing. And so that's shown, I think it's like maybe a brownish purple-ish color or purple color on the graph. And then, of course, you have the clinical symptoms of dementia starting around the MCI dementia phase, which is shown, and that's a brown line. So there have been a number of genes identified as either causative or risk factors for Alzheimer's disease as well. So we have not only protein biomarkers, but we can also look at genetics. So a large number of these genetic risk factors have been identified from genome-wide association studies, or GWAS studies. And so what's shown here in this figure is what's called a Manhattan plot. It's named that based on the idea that it looks like a Manhattan skyline. So on the x-axis, or the bottom row, you have the chromosomes, and on the y-axis, you have significance. And so if you look at the dots that are at the top that kind of look like the skyscrapers or peaks of the Manhattan skyline, these are actually genetic variants that were significant in the analysis. And this analysis was looking at people with Alzheimer's disease relative to people who are cognitively normal. So this is actually a very large GWAS of around 39,000 people with clinically diagnosed Alzheimer's disease, about 47,000 people with proxy Alzheimer's disease and related dementia cases, and just over 400,000 controls. So this study actually identified some of the same genetic risk factors as previous GWASs, such as ApoE, but also identified an additional 42 different loci that were significantly associated with Alzheimer's disease. So similar to the trajectories we see in the previous slide, these genetic markers were largely defined in a European population. So the same question is, how do these risk factors, not only the biomarkers shown on the previous slide, looking at amyloid, tau, neuropsychological assessments, but also genetic risk factors? How do all of these biomarkers and risk factors, how does those trajectories look in the Black community, considering what I've shown you so far in the past two slides were identified in white people? So I wanted to talk about some work from Dr. Elizabeth Rose Maeda. She's also a professor here at UCLA in epidemiology. And this is actually showing the incident rate for dementia across different ethnic and racialized groups. And this is using data from Kaiser Permanente in Northern California. And you can see that people who self-identify or have been racialized as African American or Black, in the very light blue on the left-hand side, they have the highest incident rate of dementia relative to other ethnic and racial groups, including the white group, which is the darker blue, and it's about 19.3. So the fact that most of those established risk factors for Alzheimer's disease dementia that I mentioned in the previous slides has been identified in white individuals is an issue. This is an issue considering Black individuals are at greater risk, are at a higher risk for dementia. So I also want, in line with a lot of health equity researchers, I wanted to build upon something as well that Dr. Maeda uses here at UCLA and focusing on these health equity research goals. So ultimately, we aim to identify drivers of racial, ethnic, and other social inequalities in health to inform policies and programs to promote health equity. There are current research efforts, though, focused on describing health inequality by group and determining if these inequalities are larger or smaller over time. And furthermore, we also want to evaluate specific risk factors and their contribution to health inequalities is key. As I mentioned in the previous slides, because one size does not really fit all when it comes to biomarkers and trajectories. And so, and by that, I want to refer to the notion that, these risk factors were identified in white populations and may not have the same effect in other racialized groups. So I'd like to start with an example of why it's important to focus on identifying risk factors in non-white communities. So this plot shows global cognition on the y-axis, and so higher is equal to a better cognitive performance. And then on the x-axis, you see age, or which you can consider age time as well. So this is going about every 2 years from the ages of 68 to 90. And we can actually see two things here. First, at baseline, which is highlighted in green, so that baseline being cross-sectional or just one time point, we see that Black individuals, which is the dotted line, gray line, have lower average cognitive performance relative to white individuals. However, if we look longitudinally, so over time, not cross-sectionally, we see that the slope of cognitive decline is actually not different between Black and white participants. And I have this little animation here to kind of, so you can compare a little bit better, and I'll do that again. So this draws our, highlights two really main important effects. First, that there are racial bias that exists in neuropsychological exams. So neuropsychological testing was created and validated by and in white individuals. So every group, every other ethnic and racialized group that is not white tends to have a lower intercept of cognition relative to white individuals. So this is an issue, right? When we are using neuropsychological testing in order to classify a person as having dementia or not. And so if we're using one time point and we know that Black individuals have this lower intercept, it's really important to remember that, to take into account some type of normalizing process in order to try to understand what that score truly means in Black individuals. And it also highlights the need for longitudinal studies in Black cohorts, because again, if we look at the slope between the two, they are identical, which does suggest that the neuropsychological test is measuring the same cognitive function in Black as it is white individuals. However, longitudinal studies is really needed to see any of that effect. Next, genetic risk factors are also not the same in Black individuals relative to white individuals. So on chromosome 19 lies two different genes that confer risk for cognitive impairment and Alzheimer's disease. So first is the apolipoprotein E, or ApoE gene. And the ApoE gene has three alleles, E2, E3, and E4. E2 is associated with lower risk or may be thought to be protective against Alzheimer's disease. E3 is present in the general population, and E4 is associated with increased risk for cognitive impairment and Alzheimer's disease. And it does have a dose allele, so having two E4 alleles is associated with increased odds for Alzheimer's disease relative to having one E4 allele. However, so you might think then, okay, E4 is bad. So if you have more E4, then you have a higher chance of developing dementia. So if we look at Black individuals who are cognitively normal, that is the solid red bar in this graph, we can see that this group actually has a higher frequency of ApoE4, and that's on the y-axis. So Black cognitively normal individuals have a higher frequency of ApoE4 relative to white cognitively normal individuals, which is shown in the solid blue bar. And the pattern bars next to the solid bars are those with Alzheimer's disease. So if you look at the red and the blue pattern bars for E4 frequency, you see that they're almost identical. So if E4 is supposed to be this great genetic risk factor for Alzheimer's disease, then shouldn't the pattern bar for Alzheimer's disease for Black individuals be higher? But it's not. So this is really suggesting that there must be some other factor than E4 that is actually contributing to AD risk in Black individuals. And this is true. This has been seen in a lot of different studies from other groups that E4 has a reduced or mitigated effect on cognitive impairment and Alzheimer's disease in Black individuals. And just to kind of show a little translational aspect of this research, there was a group that also looked at E4 expression based on ancestral background. So people have admixed genetic background, especially Black individuals from America, due to transatlantic slave trade, atrocities that were done to our people who identify as Black in the United States who have been here for generations. The genetic background is highly admixed between African and European ancestry. So that means a person who has an ApoE4, for example, may have gotten that E4 from their African ancestral side or from the European ancestral side. So looking at E4 expression based on African or European ancestry, we actually see that people with E4 and African local ancestry actually have a lower E4 expression, which is that purple, relative to people who are E4 with European local ancestry. You can see a lot more purple here, meaning there's a higher expression of E4. So this is really interesting, again, just kind of showing here is this really well-known genetic risk factor for Alzheimer's disease, yet it is not having the same expression on people of different ancestral backgrounds. Just upstream of ApoE is another gene called TOM40. TOM40 has a specific SNP, or single nucleotide polymorphism. It's that RS523 number there. And this codes for T's. So at this location, you just have T's, just a lot of T repeats. And if you look at the figure that's there, this is just a histogram showing the count on the y-axis and the TOM40 length on the x-axis. And you can see that in general, people with this specific polymorphism have T's between around 15 to 40. Often, TOM40 has not really been studied a lot in the past because TOM40 is in linkage disequilibrium with ApoE, meaning that it's like inherited together. And so because ApoE has such a strong effect on Alzheimer's disease, it was thought that the effect that people were seeing from TOM40 was really just the powerful effect of ApoE. So, of course, most of these ApoE and TOM40 studies were done in people who identify as white. And if you look at TOM40 in people who identify as Black, you can see there doesn't appear to be much of a difference. However, if you look at this by APOE background or genotype, you can see that if you're white and E3, you have poly-T repeats less than 20 or more than 30. And if you're E4 and 520 E4, you have poly-T repeats between 20 and 30. So they call this short, long, and very long. Now, if you're Black and E3, you have a very similar variation of short and very long. But if you're Black and E4, like, wow, the variation is just striking, right? It's pretty much all over, short, long, very long. This is very different than white E4 carriers, and it has, and this variation has yet to be really investigated with other AD phenotypes to see what this might mean for Black E4 carriers. So for this study, I used data from Rush University, and Rush University has 3 amazing aging cohorts that enroll at age 65 or older. And without known dementia, so they can follow them over time. And these 3 studies are the Religious Order Study, the Rush Memory and Aging Project, and the majority of the Black participants come from the Minority Aging Research Study, which includes Black individuals from Chicago and the surrounding suburbs, of which Dr. Lisa Barnes at Rush is the PI on. They also have a very in-depth neuropsychological battery, and so looking at all these different cognitive domains, such as visual-spatial, language, episodic memory, working memory, and all of these different cognitive tests are aggregated to make a measure of global cognition. So we wanted to look at, again, at that variation of Black E4 carriers in TOM40, since there was a lot of different poly-T repeat lengths. So for statistical sake, we had to group these somehow. So we decided to group them based on the short allele. So if you have 2 shorts, this is represented by red, and that's the same color in the figure to the right as well. If you have 1 short allele, that's represented by blue, and if you have no short allele, that's represented by gold. And so if we're looking at the figure on the right now, we can see on the x-axis, we have time in years. So on average, these Black E4 carriers had about 8 years of cognitive follow-up. And then on the y-axis, we have global memory. And again, higher is equal to a better memory performance or better cognitive performance. So we can see that if you were Black E4 and did not carry the short allele, which is represented in gold, you actually had a significantly faster rate of cognitive decline relative to if you were Black E4 and did carry a short allele, represented by the red and blue lines. Now, remember, white E4 carriers have no variation. They're only represented by 1 line, which is the black line here. We can see what I talked about earlier, and that is if you look at just time point 0 cross-sectional, it looks like overall white E4 carriers have this better memory performance at one time relative to all of the Black E4 carriers. So this is very similar to what I talked about at the beginning of the presentation. But when we look again at the slope over time, we do not see a significant difference between white E4 carriers and Black E4 carriers who do not carry the short allele. Where we do see a difference, though, is between white E4 carriers, the black line, and Black E4 carriers who do carry a short allele, which is represented by the blue and red, such that these Black E4 carriers who carry short are just showing a slower rate of cognitive decline relative to the other groups. So when we're starting to look at Black E4 carriers in our other studies, if we're not taking into account other genetic variation that is specific to them that may be a risk for cognitive impairment or dementia, it may actually be, muddying up our analyses a bit. And I think it's just also important to know that there are very other non-APOE genetic risk factors that have been identified in Black individuals. This is data from a recent GWAS, Genome-Wide Association study, that identified new common loci and novel rare loci in Black individuals. And then also, I think it was really interesting, again, if we're focusing on people of ancestral background, there were 2 different loci that actually conferred risk and protective effects of dementia. This pink line, I don't have the exact SNP reported, but this pink line conferred lower AD risk for Black individuals. So let's better, lower risk, while the black line actually conferred higher AD risk for Black individuals. So again, there are other genetic variants that we should really be accounting for when we're looking at dementia risk for Black individuals. And I have been talking about genetics, race, and genetic ancestry a little bit in these slides. And so I think it's really important to understand the terms before diving more into the research. Unfortunately, people have inappropriately correlated race with one's biological background. Race is not biological. It is social category forged historically through oppression, slavery, and conquest. However, race may capture some important epidemiological information, such as socioeconomic factors, racism, discrimination, and factors that heavily determine from structural racism and discrimination that led to systemic policies such as redlining. For example, race, or more appropriately, racism, can influence every aspect of life, from healthcare to education, green space such as parks, and other community resources. Genetic ancestry, on the other hand, correlates more so with geographic location, since the catalog of genetic diversity is gathered from people who live in one area, which also leads to a question that I won't be exploring today, but that is genetic ancestry. But is genetic ancestry also a reflection of environmental factors and potential epigenetic changes? Additionally, while there are folks out there working hard to change this, there is a lack of genetic diversity in reference panels that are used to calculate genetic ancestry. In fact, GWAS catalogs are roughly 80% European ancestry and only 2% African American ancestry. So understanding the difference between race and genetic ancestry is going to be crucial for the ethical research in the Black population. Yet another difference is postmortem neuropathological insults are not the same in Black individuals. So here's a study by Dr. Lisa Barnes at Rush University, and AD in blue just means that pure AD pathology, that is these amyloid and tau pathologies I talked about earlier. And as you can see, very few people with AD just actually have amyloid and tau tangles. And in fact, less Black participants have that true AD pathology relative to white participants in that the blue wedges. So in fact, Black individuals just have a much higher rate of comorbidities and other pathologies that may be contributing to overall cognitive status and dementia risk that we are overlooking at this time. And so just further going to an example of that, you can see just by looking at the years here for recent studies that have looked at protein biomarkers, it's been very recent. There hasn't been a lot of work done in this area. And one thing that is noticeable, though, is that there is discordant amyloid findings, whether people found higher, lower, or no difference in amyloid between Black individuals. So this leads to yet another study that I did while I was at Stanford University with Dr. Beth Mormino, and that is looking at people with in the preclinical Alzheimer's disease stage and using amyloid PET to look at the trajectory of amyloid in these individuals, or the levels of amyloid in these individuals. So for this, I actually used data from the anti-amyloid and asymptomatic AD, or A4 study. So on the x-axis, you have APOE genotype by a racialized group, Black in red, and white individuals in blue. And then on the y-axis, you have a amyloid levels. So higher amyloid is equivalent, is worse. So just by looking overall, you can see Black individuals have lower or better levels of amyloid relative to white individuals. But what's interesting is that this lower levels of amyloid is only significant in Black E4 carriers. So again, we have amyloid, which is supposed to be this well-established risk factor. We have E4, that is supposed to be this well-established risk factor for dementia and Alzheimer's disease. Yet, now we have shown multiple times, not only with the Tom 40 APOE study, but also here, that here are that they're not having the same effect in Black individuals. So if E4 is not conferring the same risk, what is, right? We need to be expanding beyond these well-established risk factors to look for potential effects. Additionally, looking at African ancestry. So again, I talked about how people who are Black in the US have a highly admixed genetic ancestry between both European and African ancestry. So shown here is levels of African ancestry in red, the red asterisk, and the blue asterisk, the blue star, is levels of European ancestry. And so each person has five stars to represent the 5 major groups that are shown at the top of the graph. So if you look at just the very first column, you can see that we'll just call it participant 1 has an African ancestry of around 28% and European ancestry of around 90%. However, this person does self-identify as Black. So you can see that African ancestry exists on this continuum. So I really wanted to look at that as well. So again, I'm showing increasing levels of African ancestry on the x-axis and amyloid on the y-axis. And again, higher amyloid is worse. As we see, with increasing African ancestry, we have decreasing amyloid, aka better. And this effect was also independent of amyloid. So now we have another factor that's contributing to potential protective factors against these well-established AD risk factors. And another thing that's really important, though, is looking at selection bias in these studies. Remember, I talked about earlier how the neuropsychological test was validated, creating white individuals. People who are Black always have performed at a lower, aka worse level relative to white individuals. So could there be bias into who's actually getting into the study? And indeed, we see that Black individuals are more likely to screen fail and be kicked out of the A4 study. And why? Because Black individuals have too low of cognitive test scores. So the norming procedures that are in place right now to try to close that intercept gap in neuropsychological testing is clearly not working, because Black individuals are still more likely to get kicked out of these studies versus white individuals who are more likely to screen fail out of the study due to having too high of cognitive test scores. And then I'm also just going to briefly talk about, I've talked about this amyloid imaging, but also it's really important to also note that there are MRI measures of brain aging. So for example, I talked about there being atrophy in the medial temporal lobe, amongst other areas in the brain. But what Dr. Indira Turney had showed from Columbia University in people who identified as Black, if you can look at the lower left figure there, what this shows is that there are Black individuals were more likely to have earlier brain aging relative to other groups. So you can see that the two lines are kind of like at a slant, showing these significant differences relative to the other racialized and ethnic groups. And so brain aging occurs earlier in Black individuals. Therefore, us having these cohorts that start at age 65, we're probably missing a lot of different factors contributing as well. And so this all ties back into the health equity research. What can we do better? How can we better service the Black community when we are doing our research? And an example of this is really focusing on the health disparities research framework. So let's not just focus on the biological factors, which I have been to date, but let's also focus on environmental, sociocultural, and behavioral different factors that might contribute to these. So, a person doesn't just wake up at age 65 and then they start to have memory decline. These factors occur over the life course, and it's really, really important that we start collecting data or asking questions about a person's life. And an example of that is from Dr. Crystal Glover, also at Rush University, who organized the different health disparities research framework questions into how does this associate with perceived stress? And I covered up the ones that were not associated with perceived stress, but this is a way that you can look at different factors and how it associates with a something of interest, perceived stress. Additionally, something super important that I'm very interested in, will be pursuing here at UCLA, is looking at structural racism and how that and discrimination and how that drives health disparities research. So Dr. Paris Adkins Jackson also at Columbia University has really really great papers that are looking at how to use structural racism and discrimination appropriately and ethically. And it's important to, how does this tie in to everything we're doing? When we talk about the life course, a person experiencing racism, again, that's usually something that has happened over the life course. Experiencing racism or discrimination is extremely stressful, This causes a lot of different changes in the body and may eventually cause changes in biological processes. So it is important, again, that we still focus on tying in these social factors. And here's an example of that as well, from Dr. Laura Zahadny, that show that discrimination and racism are both higher levels of discrimination and racism are associated with more hippocampal atrophy and higher white matter hyperintensity volume. And this leads to the final goal of the day, and that is just implementing health equity frameworks to enhance participation of historically excluded populations like people racialized as Black. Oftentimes, racialized groups are blamed for lack of participation in research studies, when in reality, it's the institution or the researcher who has not put in the work to target these communities. Again, I go to the phrase like one size does not fit all. And I myself have heard it's too hard one too many times when referring to recruitment and retention of racialized communities. While I haven't actively done this work myself, and I plan to in the future, I'm thankful for researchers, as shown here, for people who have done the work and put these publications out there in order to help people such as myself who may want to do research in the Black community have some type of start on how to do inclusive research for the Black community. So this really includes focusing on inclusion and exclusion practices of research participation targeted toward a specific community. And an example of this is shown here by that model with this research participation, and this is called the Participant and Relationship-Centered Research Engagement Model. So this is just one model, but it is extremely important, again, that we really focus on health equity frameworks in order to best service historically excluded, straight up excluded populations. So again, why research within the Black community is important, within this community. Black individuals have a higher rate, are more disproportionately likely to be diagnosed with dementia, yet whiteness continues to be centralized in research. Again, I'm talking about this GWAS catalog that shows about 80% of European ancestry in the blue, while if you look at the orange little wedge on the bottom right side, only 2% is African. And this is really important because the United States is becoming more diverse. This is data from the census, and you can see for the first time that people who identify as white in the United States is actually on the decline, while all other ethnic and racial groups are on the incline. So it's going to be really, really important that we bring these health equity research frameworks to non-white communities, including the Black community. And this leads to my research at UCLA now, and this is really looking at the intersection of biological and social factors, how it impacts brain aging and cognition and Alzheimer's disease. And I am new to UCLA, so I don't have a lot of, haven't shared any research that I've done here because it's still ongoing and not published. So a lot of this work that I showed was actually done at Stanford University with Dr. Beth Mormino and in collaboration with Dr. Lisa Barnes at Rush University. And I always like to take a moment to thank the women of color that are scientists in my life, because it is extremely hard to be a woman of color in this field. And without their support, I would not be where I am today. And so with that, I would like to thank you for listening, and I hope you enjoyed the lecture.